Hello guys, Zia on this side. Let's start with a brief introduction of mine. I've done MSc Zoology, have qualified CSI and JRF with All India rank 65. Have qualified set Maharashtra as well for the lectureship and one more exam that is called as PET for the PhD. Currently I'm doing my PhD at SPPU Pune. Now, let's start with our today's topic. Ultimately, like the topic which we will be dealing today is called as DNA fingerprinting. It's called as DNA fingerprinting. Now, DNA fingerprinting is basically the method for comparing the DNA sequence for any of the two individuals. It is a method to compare the DNA sequence for any of the two individuals. This was proposed by, or this was like invented by Alec Jeffrey, or rather I should say Sir Alec Jeffrey in the 1980s. Now, here let's start with the topic. 99% of the base sequence in all humans are same. 99.9% of base sequence in all humans are same. Now you will be asking, how come like the humans are looking at, they are showing a lot of diversity. So ultimately you cannot go for like the Japanese individual and the African individual have any similarity in them. But still, don't forget that they both have one single scientific name that is called as Homo sapiens sapien. So basically both of them are at the end of the day exactly same. So 99.9% of the base sequences are same, and rather 0.1% only makes every individual special or unique. Now, it is really difficult and a time consuming task to sequence and compare all 3 into 10 raised to 9 bases in the two individuals. So instead of considering the entire genome, you can consider certain specific regions called as repetitive DNA sequences. Which are used for the comparative studies. So basically you can literally use some like marker genes or something like that. Here, basis of the DNA fingerprinting, here you will be dealing with that the repetitive DNA is separated from the bulk genome since it appears as a distinct peak during the density gradient. Now, I repeat that the repetitive DNA is separated from the bulk genomic DNA because it appears at a distinct peak during the density gradient. So basically you will be having two different peaks. The major peak which is formed by the bulk DNA and the smaller peak which is formed by the satellite DNA. Hopefully you do remember about the satellite DNA which we have explained already in the chromosomal basis of inheritance, but still I am just going for a brief revision. Satellite DNA are basically non-coding DNAs which are present after the telomere. So please deal with the secondary constriction. Now, again dealing with the satellite DNA, they are of two types called as the microsatellite and the mini satellite. Microsatellite and the mini satellite. They have like the repetitive units in them, and basically they do not code for any protein. Then what is the function of satellite DNA? They have a major role to play in the DNA fingerprinting. Now let's come onto a word called as polymorphism. It is basically as a result of mutation, a germ cell mutation which will be passing into the next generation and the next generation and the next generation will be giving rise to the polymorphism in the population. Basically, polymorphism is the many forms of a same individuals. Many forms in the same individual. So basically, the inheritable mutation is observed in the higher frequency in the population is called as polymorphism. Inheritable mutation which is observed in the higher frequencies in a population is called as polymorphism. And basically, this polymorphism arises normally in the non-coding sequence. This polymorphism arises normally in the non-coding sequences because the mutation in the non-coding sequences does not affect the individual's reproductive ability. So ultimately they will be reproducing themselves on a normal basis, and that is the case they will be continuing that the polymorphism in the next generation and the next generation, and ultimately they are increasing the frequencies of the polymorphism in the given population. That is the case. Now, if I'll be dealing with the case here, how come the DNA fingerprinting method works, or what is the method for the DNA fingerprinting? So basically you have to know one single word that is called as a VNTR. VNTR. That VNTR means variable number tandem repeats. Variable number tandem repeats. These are the satellite DNAs which have high degree of polymorphism. They are continuously repeating and repeating and repeating, and ultimately they have the tendency to be in the next generation and the next generation and so on. So basically they are used as a probe in the DNA fingerprinting. They are used as a probe in the DNA fingerprinting. Now here, first of all, the DNA from the individual is isolated and cut with the RE. The DNA from the individual is isolated, which means like the nucleus will be opened and the DNA isolation will be take place, and then the DNA is cut with the help of RE, or we call as restriction endonuclease. Here that the fragments are separated according to their size and the molecular weight on the gel electrophoresis, according to their size and their mole weight. Like their size and their weight, they'll be separated on the gel electrophoresis. Here now the fragments which are separated on the gel electrophoresis are blotted. They are blotted, which means they are kept on a synthetic membrane called as nitrocellulose membrane, either or nylon membrane. So basically like the firstly what we did, we have isolated the DNA, then we made small like them, we have cut them in small fragments with the help of REs. Then those fragments are blotted on the synthetic membrane with the help of gel electrophoresis, and that is called the membrane's name is nylon or nitrocellulose membrane. Now these fragments are hybridized with the VNTR probe. These fragments are hybridized with the VNTR probe, rather I should say like these are the complementary probes which will be hybridizing. This hybridized DNA fragment can be detected by autoradiography. This hybridized DNA fragment can be detected by autoradiography, and they'll be just going for this VNTRs vary in size from 0.1 to 20 kb. VNTRs vary in the size from 0.1 to 20 kb. Hence, in the autoradiogram, there will be different bands formed, and the different sizes bands will be obtained easily. In the autoradiogram, different size bands will be obtained easily, and these bands are the characteristic for each individual. These bands are characteristic for the each individual. Now here like this was all about the DNA fingerprinting, in which like we can literally go for if we like I'll be going for the use of the DNA fingerprinting, I can literally deal with like this is basically used in the criminal scenes or the paternity disputes or somehow like the forensic sciences, because each and every one of us is having different VNTR sequences, but still only one single case has the exception, that is none other than identical twins. Identical twins have exactly same VNTR alignment. So that is the only exception. Rest each and every organism, even the children of the same parent, are having different VNTR sequences. Hopefully this lecture is beneficial for you guys. Thank you very much.